Hello, everyone. Thanks for joining. We'll get started in just a couple minutes. As people make the connection to Zoom. Hope everyone is doing well today.
Okay, I think we can get started. Good afternoon, everyone, and thank you for joining us. And welcome to the NIH Blueprint Neurotherapeutics Network Q&A webinar. We will start off with a presentation of slides, followed by questions and answers. Please submit your questions in the chat section below, and they will be answered at the end of the presentation. This presentation will be recorded and placed on our YouTube page in the upcoming days. I will be sending that link along with the presentation slides to everyone that has registered. Please remain muted for the durations of the presentation, as well as keeping your video off. I will now turn it over to Dr. Charles Cywin.
Thank you, Rakonda. Thanks, everyone, for joining today. It's my pleasure to tell you about the NIH Blueprint Neurotherapeutics Network, BPN. I am the Director of the Small Molecule Neurotherapeutic Development here at NINDS and have responsibility for the Blueprint BPN program for small molecules, as well as overarching responsibility for the HEAL Pain Therapeutic Development Program. I'm housed in the Division of Translational Research at NINDS, National Institute of Neurological Disorders and Stroke. And our goal in the Division of Translational Research, otherwise known as DTR, is to accelerate basic research findings towards patient use for neurological disorders and stroke. But these programs are also, I'm going to talk about today, a trans-NIH and have broader mission beyond NINDS's mission, and we'll talk about how that works as we go through. But the programs we do have in span from everywhere from assay development all the way through early clinical. We have funding opportunities announcements that include biologics, small molecules, which I'm going to talk about today, and as well as devices. So if you don't happen to be doing small molecules or have another project that's beyond small molecules, I'll touch a bit on the other modalities and where you can go for those. So the Blueprint Neurotherapeutics Network has been around since 2011, and we are in our 13th year of working on this, and there are many, many examples of successes from the program and have moved forward. And we work in what's called a cooperative agreement mechanism. And that means, unlike an R01 space, we work closely with you. And in fact, I'll get into this, but we meet very regularly. These cooperative agreement mechanisms are not just grant funding, but access to other resources, which I'll talk about. There are two different tracks. There's the UG3, UH3, which takes all comers. So you can be a small business, you can be an academic, you can have foreign components, you can be even a foreign institution, and apply under the BPN UG3 UH3 mechanism. Its present PIR is PIR 25-051. It replaced the prior funding opportunity because of the going to simplified review criteria, and as such, the next receipt date has two different receipt dates compared, or has receipt date different from the U44. So in January, the receipt date for this will be January 27th. The small business SBIR version of this cooperative agreement, which is an SBIR fast track application, is laid out identically in terms of programmatic execution, but is funded. If you're an SBR eligible organization, you can get funded under that. And there's some pluses and minuses about utilizing either mechanism. If you're a small business, and please talk to me more about this individually after this, and I can tell you what the considerations are for your particular situation. But the receipt date for that funding opportunity in January is January 15th. So do note there's a difference between these receipt dates. Do not confuse them, as this one will be due January 15th, or you submit it after January 15th, you may not get it accepted. January 27th for the UG3, UH3. What is our vision? Our vision is to combine the strengths of NIH industry expertise for small molecule neuroscience, drug discovery and development, together with all the resources needed to move your project forward. So we rely on NIH investigators like yourselves to initiate the ideas for novel drug targets and novel starting points for optimization. You must have the disease biology assays to drive the SAR studies if you're doing those, the animal models and SAR and disease biology assays to characterize any hits or leads that you might have down the road. That has to be coming from your grant budget. Everything else could come from NIH resources outside of the grant through the cooperative agreement mechanism, or we can mix and match to what you actually need. But what we do is put you with advisors. We have hired industry consultants to work in and with our teams and guide them. And the purpose of that is to give them expertise in medchem and PK and tox, things they might not have on their team, the disease agnostic requirements for moving drug discovery and development. And that's important to moving things forward. So the goals in all cases is to get to the clinic. It's a milestone-based program designed to de-risk the project from wherever you start to wherever you exit, to improve the ability to get it licensed and get external funding to take it farther. We would like the projects that go through this program to be picked up and continued by people with deep pockets that can afford to pay for phase 2 and phase 3. But obviously, in some cases, that may not exist. You might have to do additional grant funding or other means to move forward with the project. But we hopefully de-risk it to the point where that's possible to do. We want to invest in by identifying the best ideas for translational research from the NIH community, and this is done through this funding opportunity by doing reviews of it, due diligence, and really determining which project we're going to start. We provide non-dilutive funding in terms of the grant funding and the necessary resources in terms of contracts and consultants that are typically lacking out there. The overall arching goal is we're trying to preserve your intellectual property so that you have unencumbered IP in order to do licensing and make the program be successful ultimately. The minimum criteria of coming in is you have to have a project within the BPN mission. So there's a number of different institutes and centers that are signed on. It has to be in one of those institutes and centers mission in order to come in. You have to have a strong premise for that indication area and develop your target product profile accordingly. Look for a starting point, chemical starting point, whether that be a literature starting point or whether that be something from a screen. You have to have the chemical starting point in hand and have, as I said, the SAR driving assays in your side to move it forward. And from the contract side, we can provide all the other assays like in vitro ADME and so forth can come from additional contract work that we have. Our network is set up like this, where we just create a lead development team, which includes you as principal investigator, those consultants we mentioned, and NIH staff. The NIH staff are there to facilitate, and then what they're facilitating is this team functioning well with all the moving parts, the grant dollars, the contract, the NIH contracts that we have put in place on your behalf, as well as the consultants we give you access to. The NIH staff is the ones coordinating all that and making sure that you get what you need when you need it. It's tailor-made to your project. It's non-dilutive, as I said, protecting your IP. The thing to ask yourself is, do you have the expertise and resources to do these activities readily in-house? I get a question almost every time about, can I do X myself? And my answer to that is always, it depends. Do you have the people already in place to do it? If you do, then perhaps you can. Because this is a milestone-driven program, it can end abruptly. Every year milestones are evaluated and funding continuation is determined based on availability of funds, meeting your milestones and program priorities overall. And so projects can and do stop for scientific reasons uh, fairly often. And when you do, you don't want to have to get rid of the person you just hired to do something. So that's a way to manage it by us having these contracts. We have med chemists available. You don't have to hire them and then get rid of them if something goes awry. Tox and drug manufacturing formulation, I wouldn't expect you all to have, but and it usually comes through our contracts. Likewise, a clinical trial piece for healthy human subjects, a phase 1 clinical unit, is typically not something most people have readily available. They contract it through us in most cases. Everybody's doing lead optimization will get access to our relational database system, which is presently CDD, and that allows us to put all the data in one place. You will be asked to use BPN codes for all your compounds for clarity across all the projects. Whose institutes are assigned on, which institutes are assigned on, who's the contact for them is listed here. So NINDS, that's me. NIA, alcohol abuse and alcoholism, National Center for Complementary Integrative Health, NIMH, DCR, the Eye Institute, drug abuse. They all participate in this program. There are some restrictions, and not everything not every IC accepts every kind of project. So I'll talk about that again in a moment. All projects enter at the preparatory phase, and the preparatory phase is really just that first year in most cases that we're talking about. So the UG3 or the U44 phase. You enter there trying to get to the pieces that you're missing, or if it's early stage, completing the early milestones that are required to sort of kick the tires, make sure everything is as stated, and get you up to where we expect you to be meeting the milestones as we progress through. The progression is a administrative progression. So when we decide to go from UG3 to UH3 or U44 phase 1 to U44 phase 2, you don't have to go back to a whole review panel. It's the administrative review within NINDS and the programmatic staff, as well as the NINDS director or the director of the other IC if it's being funded there. You can only receive one UG3 under the grant, so I can't like extend the UG3 a second time. You would have to apply for a new grant if that was the case. We typically support up to 1 year of medicinal chemistry in exploratory phase, and maybe 2 more in the maximum in the execution phase. That'd be like 3 years of total med chem effort to do optimization, and the last 2 years at a maximum in that scenario are devoted to the pre-development and development activities required to get the IND filed and start the phase 1 clinical trial. As I mentioned, all projects come in preparatory phase, but we have both. You can enter anywhere along the path, discovery or development. However, not all the institutes and centers accept development projects. For instance, NIDA, the National Institute of Drug Abuse, and NIMH, National Institute of Mental Health, do not accept development projects. So if you have a development project and you want to come to BPN, you're going to be out of luck for those indication areas. You'll need to go directly to those ICs and discuss it with them what funding mechanism they would prefer you to use. In the funding opportunity announcement, and this needs a correction. I always have something I forget to correct. This is 25051 now. And in the funding opportunity, it'll have a link to the milestone orientation. It's also on our website that I asked Rakonda to put in to the chat. So the chat and the funding opportunities are in the chat, both the link to the website and the funding opportunities, including the correct one, 25051. And you'll want to look very carefully at those funding opportunities. They have some requirements, but they also have a lot of information in there, what you need to do. The budgets, you need to propose a budget that you actually need. It should, in the UG3, UH3, it should not exceed $499 in direct costs in any year of the grant. So, and you must submit all years of the grant with the grant application. So you're not submitting just a grant application for UG3, you're submitting a grant application for UG3 and UH3 or U44 phase 1 and phase 2. But you're allowed up to, 300K in direct costs on average, sort of a typical UG3 phase, and up to 1.5 million direct costs is theoretically possible in UH3. You would have to ask permission to exceed that 499.9 that I mentioned in at least 8 weeks in advance if you need to do that. This would only come into event if you had a pre-existing relationship with a contractor that it didn't make sense to use the NIH contracts to go instead. And there we might consider paying this. Uh, the, the majority of the money in these projects are really in the contract side as you progress to the development phase where things get very expensive very quickly. As I mentioned, there's some differences in the budget requirements. The U44 version follows SBIR rules. This is NINDS rules. You need to check your specific IC. So if you're coming from NIMH or NIDA, they all have different budget restrictions for their SBIR program. You need to check with the SBIR folks in those institutes to know what those are. But if you have a vertebrate animal section, you could be up to like 500K, 700 across 2 years, or 1.5 million up to maximum of 3 years, $3 million in the phase 2. So one of the differences here, as you can see, is most phase 1 or UG3 are going to be 1 year. That's what the expectation is. And so in the SBIR, you'll only get typically 4 years total funding in that scenario. The reason I say that is, 700,000 across 2 years in total cost is not sufficient to run a program doing lead optimization work or hit the lead work even in there. So that's why you'll typically have less years in this versus this program. So 5 versus 4 in most cases. You're probably going to need the 5 for many of your projects. So do consider this. The disadvantage is related to having to ask permission to go over versus just you can submit it directly based on the total dollar cost limits here. You can't charge a fee where you can charge a fee on SBIR. You get maximum indirect rates without a negotiated rate agreement. It's probably going to be 10% versus, I think, 40% on the U44 version. So those are things to think about in the gestalt of everything to make your decision. But first and foremost, make sure you're eligible for an SBIR if you're going to apply for an SBIR. And many, many people can't meet the rules. So do look very carefully. I also get this question all the time about foreign applications. I mentioned the UG3 UH3 mechanism will accept foreign applications. It will not take it from the U44. That's SBIR, and therefore foreign applications are not eligible. But for the UG3 UH3, they are. But the reviewers will assess the project whether the project presents special opportunities for furthering research programs that through the use of unusual talent, resources, population, or environmental conditions that just don't exist in the US. You need to recognize and explain why that would be. And it also goes to the review panel like that. And it also has to go through our council for another review separate from the funding decision, whether we recommend funding or not, to see whether we want to fund a foreign application. So there's definitely more hurdles for a foreign application. So if you're working with a US organization, you may wish to have the US organization be your lead and the foreign institution be the foreign component. Entry criteria, as I mentioned, disease biology, you need a novel target, strong biological validation, and a feasible path to the clinic. So looking at the programmatic, your target product profile, are we going to be able to if we get a drug like you say that profile looks like, is it going to be something that the clinicians will want? Um, so talking to a clinician is not a bad idea up front to help develop a target product profile, understanding what's important, what's not. Does it need to be an oral drug? Does it need to be an IV drug? Um, can they take it 3 times a day, or is it only going to be once a day? How long are we going to have to deliver it for? Is it going to be multiple days or just a one-off drug? All those questions play into the actual planning for the program. You have to have your assays in place for optimization. And so in this case, we always talk about having a primary screen, a selectivity screen, a counter screen, just to show that the assay is functioning correctly. We talk about having cell assays and also in vivo animal models if they're appropriate. Your compounds must require medicinal chemistry to optimize them if you're in discovery. That's sort of the biggest thing. If you're doing optimizations, you're still in discovery. If you picked your candidate, you're in development side of the equation. But if you are doing anything, you have to have obviously a free path to get to intellectual property that you can license. I often get people who come in with other people's drugs and say, well, I'm just going to utilize this. And if you don't have the permission from the people who own the IP, you can't do that. If you come in and say, I want to make the next X inhibitor for this target, and there's 1,000 other patents in that area, you need to do 2 things. You say, why am I going to be successful when the other teams, other people working on this with experience equal to mine have failed? You also need to explain how you're going to develop intellectual property in a very heavily mined area. So those are things to think about in your application that will make it a stronger application if you deal with it up front. The development phase is all about how strong your data package is, linking it to the disease indication. It's going to be getting to more of those disease agnostic things. You're going to have, I have my efficacy. What is my therapeutic index look like? What's the ADMET look like? Is it distributed well? Is it in the brain? Is it have a reasonable half-life, et cetera, et cetera. Again, same thing. How feasible is your path to the clinic? Can I execute a clinical trial down the road? Is the IP free of obvious roadblocks that are going to prevent you from moving forward? This is the milestone progression. Here's the link for the orientation I mentioned to you. And it is certainly something that you should read through if you have not already. Oops. And it talks with all of these things in more detail, sort of more granularity of what is expected for a milestone, more or less, for each one of these stages. These are the generic milestones for the program. Your program may have some more specifics related to your disease biology. But on this orientation slide, for each stage, it goes through those milestones. And on the right side of the slide, it has, here are the resources available that NIH will give you in terms of contract resources outside of your grant budget. Grant duration is a maximum of 5 years. Some of this may occur after 5 years. Like the clinical trial may happen after the grant runs out. In some cases, if everything goes well, even in some cases, that's the case. I started out saying we try to protect your intellectual property. Inventorship is always determined by U.S. patent law. And again, you can't short shift the law here. If someone's an inventor, they're an inventor. That's why our contracts have intellectual property clauses, and we ask you to sign intellectual property agreements when it's appropriate to do so. NIH is not seeking a stake in your intellectual property. As I said, myself and the staff are not looking to be actively determining or doing inventive things with you. We're here to help facilitate your program, keep you on the path to meeting your milestones, and we want you to succeed in getting this program to a point where it can get licensed. As such, we'll ask you if you have multiple different sites working with you, you have to have IP agreements. And there's a request in the funding opportunity that says you have to have an IP strategy. In there, it's appropriate to say, I'm working with X institution. We have an agreement in place. We're going to be the lead on the filing of all patents and so forth. Any royalty agreements and so forth with those people, you need to sort of explain up front. In our contracts, we don't ask you to provide royalties to our med chemists or our drug manufacturing formulation. The consultants, we say, you can do whatever you would do with any other consultant you hired in your organization. And we don't, again, are not looking for you to make any particular arrangement. That's not, we stay out of that. IP agreements should aim for unencumbered IP, though, consistent with the program goals. Here's our website. It's a wealth of information. It's in the chat right now. Up here at the top, you can go to BPN Biologics or MedTech to learn more about those programs if you have a biologic or a device you wanted to bring forward. The frequently asked questions has things like the link to that milestone orientation as well, but also a lot of questions that may or may not come up today. The resources talks about all the consultants and contracts we have, what they are. The External Oversight Committee is who we have that helps us review your milestone progression every year. They give us their people with years and years of experience in drug discovery and development, and provide their individual feedback to the NIH about your progress. They're not voting up and down. It's not a collective vote. They don't make the final decision, but they are giving us their feedback and recommendations for you. So the program is, as I said, been around for 13 years. These are some of the projects that have progressed into the clinic. And some of these are from the beginning of the program. Some of these are from midway through the program. But you can see at this point, we have 4 assets that started in BPN as discovery projects, exploratory discovery projects in most of these cases, and are now in phase 3 clinical testing. We have other projects in phase 2 and 1, and they're moving all projects forward. At least 10 of these programs of the projects have announced additional industry funding since utilizing the BPN, which is a substantial percentage of projects started with BPN. If you don't have all the preliminary data you need, there is a program in NINDS's world. There are similar type funding mechanisms for different things in other institutes and centers, but in NINDS, we have what's called the IGNITE program that allows you to do things like characterize your molecule. So if you have a compound that you think is a reasonable starting point and you want to get PK on that and characterize it more fully before you come to BPN, you can do it. It's not about optimizing your molecule, it's about characterizing and moving it forward. Assay development, this is about developing an assay and running a screen. This one's about developing an animal model that you might need for your program. They're all 3-year maximum. They're all phased awards. They're all milestone driven, but there are grants up to $499 any year, maximum of $750 across all years of the grant. The next application due date for IGNITE is in February. If you need a contact, that's Becky Roof, but you can see her contact will be listed in the funding opportunities, Becky Roof and Chardell Spriggs, the 2 contacts. We have a neurotherapeutics training course that we have every year. I point this out to you in case you want to learn a bit more about doing drug discovery and development. It's a great course to go through. It's a mentored course where you present your project, you learn from those people. What was also is great is we have the course material, seminars and materials available on the website from UC Davis. And so if you need a primer on PK, there's a lecture on PK. You need a primer on tox, there's a lecture on tox. You need a primer on IP, there's a lecture on intellectual property, all available on these websites. It's a great resource to learn more from people doing the work in industry and elsewhere. Some things to do. I can't emphasize this enough. read the funding opportunity and any funding opportunity you apply to very carefully because each funding opportunity is different. As I mentioned, this funding opportunity requests tables that say how much, what you're going to do, what you're going to ask BPN to do, clearly delineated. So putting a screening funnel and saying, here's what we're doing and here's what we're asking you to do is usually a good idea. You also need to have an intellectual property agreement I mentioned as a other attachment. You need to have all your years of the funding. So not only UG3, but UH3. Read it carefully. Make sure you check all the boxes, do everything it says to do. Do not try to cheat around page limits and things like that. Because if you do by trying to make the spacing smaller and things like that, you can get your application withdrawn before I ever see it. Discuss your program, your proposal with myself or Home Institute's program directors for the area of disease that you're talking about. Contact SBIR program directors for SBIR specific issues. So if you're wondering about eligibility, things like that, talk to the SBIR contacts because they know the up-to-date rules in the SBIR space and maybe aware of changes I am unaware of yet. Put forth a really solid scientific premise by giving good preliminary data that supports your proposal, that you can address the rigor of that data, and not only of the data that you plan to create, but the preliminary data that you created and the preliminary data that you cite in the literature. Was it done in a rigorous way? Was it blinded? Was it controlled? Et cetera, et cetera. Address obvious criticisms of the chemistry and the biology. Common things that people do is, they reference a compound and it's an intellectual property, and they don't say how they're going to get from patented compound to something they can patent themselves. Or it has a structural alert in it that's clearly going to make it a toxic compound or some other issue, and they don't address it by saying, yes, we recognize this. This will be a point for optimization as we move forward in the UG3 phase of the grant, for instance. If you're missing data for toxicology and you don't have it yet, say that's what we want to acquire. We're going to make that a milestone in our first year, and then follow the regular milestones. As I said, clearly indicate what needs to be done as part of the grant versus what needs to be done by BPN contractors. Don't just hide it in your justification in your budget. Make it clear in the research strategy section so that every reviewer understands what you're asking for. Because things that you're asking us to do through our contracts are not reviewable. Things that you're doing yourself in the grant are reviewable. So if you say, you don't say that we're doing the MedChem through the Curia contract, for instance, that PPN has, then they'll be reviewing your strategy for MedChem. So you do want to say, I'm doing this myself, or I'm not doing it myself. Include budget streets. I can't say that enough. Every grant cycle, I find people submit grants without all years of the budget. I try to catch them as quickly as possible so they have a shot to put it back in. But many times it's not just the years missing, it's the research strategy is missing the explanation of those out years too. So please make sure you address the whole program. I talked about rigor and preliminary data. There's a data safety, data management and sharing plan that's required for all projects. You need to explain in a drug development world that you will be publishing as soon as you can after patenting and protecting and so forth, in the publishing of the patents for licensing point of view. So you need to explain that all. Some additional things, complete your registration in 6 to 8 weeks. So now, if you haven't done your registrations for filing a grant application, now is the time. If you haven't renewed your sam.gov registration and it's due at the end of the year, now's the time to renew it before you run into problems. Consider submitting your application early, a day or 2 days before, and letting me know that it's submitted. I try to look a couple of days before. I say this because if I find an addressable problem, like you left out something you're supposed to include, or you missing a year of the budget or something like that, I can contact you and say, you might want to pull the application and resubmit it with the corrections. if you're applying for, anything that exceeds $499.99 in direct costs in any year under the UG3H3, you have to ask permission 6 to 8 weeks in advance of that receipt date. And I would say get it to me early as possible because I'm going to need a lot of documentation about your budget as well as your research strategy to support this. Talk to your tech transfer business development group at your side because you need to have good plans for that funding, for the patent funding and licensing activities to go into that research patent strategy, other attachment. Do not plan for this to be your sole source of funding for your lab. It's milestone driven. It can end abruptly. You run into a brick wall and things stop, especially in the later stages of the program. You've just run into some unexpected tox finding or something. Things can abruptly stop, and you don't want to. And as I said, you don't want to hire someone and you only have to fire that same person. If it's something that can be done through our contracts, you can mitigate the risk in those situations by utilizing our contracts. Don't under-resource. If you need more budget than the $499.99, then you need to budget for that and talk to me. Or really don't try to under budget and expect that I'm going to be able to give you money later to do it because it doesn't really work that way. Please do not plan for BPN contracts to do disease biology. We can't do that. Do not plan for us to do one-off in vitro experiments. So if all you need is a syrup panel on your development compound, you're going to have to budget that. Even though we could theoretically do that through our contract, you need to do that on your budget because one-off things for relatively inexpensive in vitro type stuff is not something that we can budget for or even do through a contract. Government contracts take a lot of effort to put in place. It's a lot easier for you to put in your budget. Do not code your application as a clinical trial if you plan to use the BPN contractors to do that. The BPN contract will be a clinical trial contract, but not your grant. So if you have human subjects for something else, like you take human whole blood in an identifiable way and so forth, you might have human subjects you have to actually report. That doesn't make it a clinical trial, but you still have to deal with the human subject section. Even if you do plan to do your own clinical trial, it's more than likely going to be a delayed onset because you won't know everything up front that you need to put in the clinical trial. So think about whether it should be a delayed onset if you put it in your grant. Of course, you'll be asking me for an ARA, so we'll be talking about this if you plan to do this because you'll be exceeding that $499.99. I mentioned there are programs for biologic. The contacts are listed here. Lauren and Kelly are your contacts for the BPN Biologics program. It can handle antibodies, peptides, large peptides. So anything over 6-mer, we send over to the BPN Biologics for sure. Proteins, gene-based therapies, cell therapies, other emerging biotechnologies can all end up potentially in the BPN Biologics. It has a different suite of contracts that allows for more flexibility in modalities versus small molecules. It has some differences on what's required to get through the discovery phase to de-risk it. So they allow a 2-year UG3 phase as well. And again, not all institutes accept development projects the same as small molecules, but it's aligned very similarly in the program. I mentioned I have responsibility for the pain therapeutic development program that Marianne Pellimonter is the program director for. If you have something that is a pain therapy that's not opioid sparing, not mu opioid targeting, you could potentially come to this in addition to BPN as an opportunity. You can talk to myself or Marianne about which one may be more beneficial in your particular situation to come in on, but both are available to you and you have to figure out which is the best fit. This does require some work on a biomarker for clinical trials that the BPN does not require. So we're ready for questions now. I'm happy to take as many as we can in the remaining 20 minutes. They can be on application process, review process, proposals and budgets, contracts, intellectual property, any of the above. Here's my email address. Feel free to reach out to me. Rakonda manages my calendar and ultimately will be one if you need to talk to me to schedule you on my calendar somewhere. So you have Rakonda's email. You can also reach out to her to get on my calendar directly.
Thanks, Chuck. And for those that may have missed it, you can submit your questions in the chat feature below and we will answer them. And yes, this was recorded and I will be sending this presentation along with the slides out sometime next week.
So any questions from the audience?
Okay, I have a question. I'm just going to kind of explain my situation. So I'm an academic clinician that basically through clinical practice has discovered that a very old generic central nervous system drug that does not readily cross the blood-brain barrier can be dosed very effectively intranasally to cross the blood-brain barrier. It's the route and all that has already been patented by my academic institution with an exclusive licensing agreement back to myself. We've had a pre-IND meeting to discuss what with the FDA, and basically they want nasal toxicity studies because of course that was never done with the actual medication, but systemic and brain and all that has already been done. So through the SBIR though, part of the requirement was that the small company do 40% of the work, et cetera, where essentially with the BPN, I mean, the BPN would be asked to do basically 100% of that animal work for me. Would that work?
Well, let me ask this one follow-up question before I answer. Were you requested to do those toxicity studies intranasal in order to do phase 1 work and do exposures in phase 1? Or were you asked to do it longer term stuff to enable a direct to phase 2 clinical trial?
Yeah, so they didn't exactly say, but before I could proceed with any phase 1 or 2 in human subjects, they wanted at least 6 months of animal studies, shorter term in 2 species and longer term in 1 species to prove that the medication itself is not directly toxic to nasal epithelium.
Okay.
I've used it clinically for 4 years in humans without any problems, so I'm fairly confident it'll be okay, but that is a required study for me to get to the phase 1, 2, etc.
Right. So you may be a better, if the indication falls to NINDS, there's something called the exploratory clinical trial funding opportunity. I asked Wakanda or someone to look it up. I mean, I can try to look it up in a second myself. That funding opportunity would probably be a better fit for you. That allows you to do some of that preliminary work and requires a phase 2, but you can even do a phase 1/2 into that particular study. So that would give you the funding in order to go get those longer term tox studies and deal it forward. I could deal with the smaller, shorter term tox studies within VPN if you were just going to do, if it was written in such a way as that you're limiting it to the short term tox studies to do a phase 1, exposure type study, we could do that probably in VPN. But if your actual goal is to run all that package and then basically go straight to a phase 2 kind of study, then that would be more appropriate for the exploratory clinical trials funding opportunity.
And can you say again what that was, or is that what you just put there? Can you say what that other funding? Because we need to do the preclinical animal data before we can move into any animal studies per the FDA. So
Yeah,
that's
and that's part of that funding opportunity.
Okay, perfect.
It's called exploratory clinical trials, and if you give me a moment, I'll see if I can find the funding opportunity as we're talking. But
And then is that part of an SBIR or STTR in that?
it is not. I don't.
Okay.
It is a UG3-UH3 mechanism. Like all UG3-UH3 mechanisms that are out there, they're all being. I'm going to give you the funding opportunity announcement. Be on the lookout for if it updates before you apply, because the simplified acquisition or simplified review criteria are being implemented after January 25th date. So all funding opportunities are being updated that fall into that policy. So
Gotcha.
you may well see this funding opportunity updated, but it's PAR 22-142.
I just put it in the chat.
Oh,
Candler
perfect.
had,
Thank you. Thank you.
yeah,
Thank you.
Candler had found it. Thanks. Okay, Andrew, if you're done, we can move to the next question.
Yes.
Okay, scroll up. Okay, the next question, Chuck. Can the initial candidate be screening hit that needs more optimization through an additional analog by catalog screen done in the discovery stage?
Absolutely. That's exactly what one of the components that we do in the exploratory phase. We do SAR by catalog. We resynthesize the hit and make sure we can reproduce the activity. In that process, we learn how to do the chemistry and start doing some analogs around it. But we also supplement that with SAR by catalog.
Okay, next question. Can you say a bit about the review process for these applications? For example, are the identities of reviewers published? Are NINDS reviewers used? What is the success rate for submitted proposals?
I can say that is a special emphasis panel done through the NINDS review branch. It is not percentiled, so they're scores. And I don't think the success rate's really much different from any other funding opportunity presently, which, all funding opportunities in the present budget climate are difficult. If you want to look, each institute has its own uh, funding guidelines associated with what we're doing. So typically, like 80% of them that are funded are scoring below 30. It may be 10 to 15% might be scoring in the 30, it may be below 32, and I have to look. But there are published guidelines for that. And if I can find it, I'll look at real. I'll put it up here in a second if I can find an example for it. But the success rate is similar, I think, to other funding mechanisms. Some people are funded in the first go around. Some people it takes multiple attempts, go through A1 and get there. But not everybody that is that applies is successful for sure. It's a highly competitive program because we're spending, many millions of dollars on your particular project. So it will definitely require a good application with good, strong scientific rationale for that.
Okay, next question. How competitive is the program? How are the projects evaluated? Same as scientific review board as a phase R43 or R44, where a group of scientists evaluate the proposals?
It is a similar science, it's a special emphasis panel, so it's not a percentile R43, R44 type grant where it might be, I don't know what those actually are percentile, but you'll get scored. So our special emphasis panel consists of drug discovery and development experts that are disease agnostic from industry and elsewhere, supplemented by every application will be for a different indication area. So we'll have additional reviewers with those expertise and for that, particular disease area on the review panel as well.
Okay. Oh, sorry.
Go ahead. You go.
Okay. Is phenotypic screening strategy compatible with BPN, or do you need to know target and MOA?
You have to be able, phenotypics is okay. And I, in fact, have and still have some phenotypic projects in our portfolio. The trick to the phenotypic is, can I drive SAR? Is it have the throughput in order to be able to optimize molecules or not? I will say there's always bias out there in to knowing more about the target and stuff and feeling because it's potentially easier to optimize on it. But a strong application explains how you're going to do the optimization in a phenotypic way is can be a very strong application as well. And I've had, as I said, I've had several score very well and be funded by us.
Okay, next question. How do investors view startup companies participating in Blueprint? Are there any concerns?
How do investors view startup companies participating in Blueprint? Usually not a problem because we're trying to maintain your intellectual property in a non-dilutive way, it's actually viewed by investors as a positive thing. In a startup world where you're a virtual company, for instance, you will probably not qualify for SBIR, and therefore you will want to come to the UG3 UH3 mechanism, and you'll look towards. I said our goal at the end of the day is to get it licensed. I have had many cases where the licensor has come in and said, hey, we have a license agreement now. Can we continue this program? And the answer in most cases is yes, that we can, and we work together and figure out what we're going to continue to pay for versus what the new people are going to continue to do. And they usually try to dovetail it to try to take as much value out of what we're doing and invest their money farther down the pike. Where it can get tricky is an SBIR grant. If you have an SBIR, the change in ownership could make you no longer SBIR eligible. If that happens, there's nothing I can do, the project ends. So if you come in a U44, that's one of those risks. Is if the day you are not eligible for SBIR, you're not eligible for SBIR, the project and the grant ends. You just can't, NIH won't give you any more money under that grant. So do keep that in mind if you're coming under the U44 and you're thinking about investors in your program. Be careful on what the impact of what does it impact your funding. Everything's manageable. You just have to know what the issues are and address them up front. Did you have any more questions? I was going to put this up just for a second.
None at the time, Chuck. No.
All right, let me just put this up because I said I would. This was the question about what's fundable. This is the guidance in NINDS. Each institute has similar kind of guidance. You have to look, but typically most applications, 32 or lower are the scores for 80% of the ones that are funded. Some applications can go in 33 to 37. Things above 37 are rarely funded, but we do have plenty of examples of, again, over 13 years I've been doing this, of doing it. Right now, as I said, the pay line for an R01 is 8th percentile, and it's 4 if you're above 500 in direct costs. The value that we give the program, not only from the grant side, but from the contract side, you can get up to, potentially 11 to 16 million dollars in total investment between the contracts and the grants by the time we're done, if you're taking all the way through to the clinic, just because of the cost of toxicology, the cost of GMP manufacturing, the cost of clinical trials. It's just super expensive. That's why the program is milestone driven. That's why, we can do this, because we move the projects forward in a very thoughtful way. Again, having the question about asking investors, if you have a separate stream of money from a separate investors, it sometimes allows you to move faster, because in the BPN milestone world, we're doing things more linearly than I might do if I had, a really long runway and plenty of money to spend. I might choose to do some stuff in parallel to move things along faster. I can't do that in the government side, because I've got to de-risk each big investment, each tranche of money as I'm going along. So that can be quite beneficial in speeding up the process. Well, if there are no further questions, I want to thank everyone taking the time, and everyone who will be listening to this after this live presentation. And if you have questions, do reach out to me, charles.cywin@nih.gov. If you need to talk to me, reach out to Rakonda and myself, and she'll get you on my calendar. And I want to thank everyone for taking the time today. Everyone have a great day.
Thank you, everyone.